Hello, this is the most recent paper for Unit 3. It is the Monday, 20th of January, 2025, and this is going to be a full walkthrough. Please, if you have any questions or inquiries, just write them down in the comment section below, and I'll try to get to you as soon as possible. Okay, let's start. Okay, so the first question is, coir fibers are collected from coconuts. The photograph shows part of a coconut with some of these fibers. Okay, so the first question is, name two types of fiber cells found in a plant. Okay, so the main fiber cells that they're focusing on is the sclerenchyma and the xylem. These two fibers are really tough and hard. Remember that the xylem is made up of dead cells, so it's going to be really hard and tough. The second question is, a student investigated the effective length on the tensile strength of these fibers. State what is meant by the term tensile strength. Okay, so now we're getting hints that they're going to be asking us about the tensile strength experiment and testing how strong different fibers are. Okay, so tensile strength is the pressure a fiber can take without breaking. I would recommend that you would just memorize this part over here, meaning of tensile strength, and also one of the core experiments for tensile strength. So I describe how to investigate the relationship between the length of these fibers and their tensile strength. Okay, so the first thing we need to do is we need to use a range of fiber lengths. So the first point is use 3 fibers with lengths of 5 centimeters, 10 centimeters, and 50 centimeters. Does it have to be exactly 5, 10, and 15 centimeters? No, it can be 2, 4, 6 centimeters. Just give them a range, okay? The fibers must be from the same plant, okay? Because it's not going to be a fair experiment if you use a range of different plants, because different plants have different fibers. Now, we need to control the temperature. So the temperature and humidity and pH must be controlled at 25 degrees Celsius. Then we're going to add 5 grams of weights until the fiber breaks, and then we're going to record the weight at which the fiber breaks. The weights are going to look, in real life, they look like coins. They just look like metal coins, and you just add them on top of the fiber every time. Okay, the graph shows the results of a similar investigation. Okay, draw a table to show all the results in the graph. So this is a 3-mark question, and these 3 marks are very easy to get. Over here on my y-axis, I have the tensile strength in MPa, and I have on the x-axis the fiber length in millimeters. I'm gonna draw a table using pencil, and then I'm going to write down the titles using pen. So this part over here is using pen, and the table itself, is using pencil. Make sure you use a ruler as well. Okay, so for the next question, they're going to be asking us about measuring the gradient and finding the y-intercept. So this is the equation that I used for the gradient, which is y2 minus y1 divided by x2 minus x1. So over here we have y2, which is 160, y1, which is approximately 200. We have x2 over here, which is 35, x1, which is 5. And then to find the y-intercept, which is plus c, because y equals mx plus c. So c is going to be simply 206. How did I get that value? I simply got my ruler and I just drew I just continued drawing the line from the main line given over here, which gave me around the value of 206. Okay, so part I, which I talked about in the previous slide, it says calculate the gradient of the line of best fit shown in this graph. Okay, so we use the equation over here, and I got my values and it gave me minus 1.33. So why is it minus? Okay, well, we have our graph, and if you notice, the line is facing this way. This is negative. The gradient over here is negative. If it was like this, it would be a positive gradient. Okay, so give the equation for the line of best fit. Okay, so this is our gradient, and our gradient represents m in y equals mx plus c. This is m, which is over here. And then our c is going to be our y-intercept, which I told you how in the previous slide to get it, which is going to be 206. Now the next question is, the drawing shows the structure of a part of a plant fiber. We have a junction where two cells meet, and we have a cell in a fiber. Okay, so this junction, you didn't learn what a junction is. However, when you look at this, you can say, oh look, there's a line. So is this line a gap? Is it something that makes it connect? Let's see. Well, usually when two cells meet, there's going to be a sort of gap in between. So we can say that it will cause the fiber to break more easily, or we can either say it's stronger. Okay, the table shows the gradients for similar graphs for three other types of fibers. We have bamboo, which is minus 3.12, brown coir, which is minus 1.25, and jute, which is minus 1.16. So state the relationship represented by the gradient. Okay, so this is all negative. So there's a negative correlation. Okay, so as the fiber length increases, the breaking strength decreases. Okay, as fiber length increases, the breaking length decreases. This question is related to the graph that we saw previously. Okay, suggest how differences in the structures of these three fibers could affect their tensile strength. Use information in the graph, the drawing, and the table to support your answer. Okay, so they are different plants, therefore they're going to have different fibers. Remember, the main part of this question is talking about fibers and their tensile strength. So bamboo may have a different fiber structure, which makes it stronger. Also, the number of junctions is different in every single type of plant that they gave us. The junctions, they may make the fiber weaker, because if it's a gap, it can break more easily. Now, question 2. Several factors are involved in the germination and growth of pollen tubes. So remember, a pollen is going to land on a stigma, and it's going to grow a tube down the style where it's going to deliver the male gametes. Over here can be our style, and then this is our ovary. Okay. So describe the role of pollen tube in plant reproduction. Okay, so they're going to release digestive enzymes. Usually at the tip over here is digestive enzymes that are going to break down the style and create a pathway using a process called hydrolysis. These enzymes are hydrolytic enzymes, meaning they use water. So hydrolysis, water, hydrolysis, break apart. Okay, so the role of the pollen tube is, number 1, releases digestive enzymes that create a pathway for male gametes to be delivered to the ovary. Keynote on delivered to ovary, that's one mark also. And releases digestive enzymes, that's another mark. Okay, a scientist investigated the effect of calcium chloride concentration on pollen tube growth. The pollen tubes were 75 micrometers long at the start of the investigation. The table shows the results of this investigation. So we have our y-axis and our x-axis. So our mean pollen tube is going to be our y, and our calcium chloride is going to be our x. Because over here the numbers are pretty stable. They're from 1 to 6. Over here there's a range of numbers. Therefore, x is going to be the calcium chloride concentration because they're stable, and y is going to be different range, which is going to make it our y-axis. Make sure when you label, this must be in pen, this must be in pen, and this drawing must be in pencil. okay? And make sure that when you're plotting the points, don't do a dot, just do an X, and do not do a star like this. That's too messy. This one's correct, this one's wrong, this one's wrong. Okay, describe how the data in the table could be obtained. Okay, so over here we're measuring how pollen tube grows in different calcium. Now calcium is required as one of the mineral ions needed for plant growth. That therefore it's also needed for pollen grains. Okay, so we're gonna always need a range of concentrations when we're talking about any substance like calcium. So we're going to need a range of 6 different concentrations of calcium ion solution that must be used. Now sucrose solution must also be available for the pollen grain in addition to the calcium ions, because sucrose, okay, is going to provide energy. Now the 6 dishes, which are Petri dishes, must be exposed to a controlled temperature and pH, because these factors also have an effect on the growth of the pollen tube. Therefore, they must be constant. The pollen is placed on 6 different agar plates. So remember that we're going to have agar jelly, which is going to contain the sucrose, which provides nutrients, and also calcium, which is what we're testing. Then we're going to use we're gonna have to leave the pollen on the agar plates for at least 1 hour to give it enough time for the pollen tube to start growing and for the digestive enzymes to start working. So we're then gonna take the pollen grain out and put it under a microscope. Then we're gonna use a microscope with a calibrated graticule to measure the pollen tube length. Okay, then we're going to repeat the readings at each calcium ion solution to calculate the mean. Okay, the following points over here must be remembered. Okay, this is a pretty standard, these are pretty standard steps, and this is a very easy experiment that must be remembered. So the way I memorized it was I wrote it down on a piece of A4 paper and I drew the steps along with bullet points, and then I just kept reading it until I memorized it. Okay, the next question is comment on the results of this investigation. Well, pollen tubes do not grow at 1 arbitrary units or 6 arbitrary units. This information can be seen, okay, from the graph that we drew up previously, where it was sort of like this. Okay, and there's also no control. Okay, if you notice on the experiment, there's no control given. Okay, so our control is going to be just sucrose alone without calcium, because just sucrose alone may have an effect on the pollen tube growth. Now, also, the relationship is not linear. So this is a linear relationship. This relationship is not linear. Okay, question 3. Photograph 1 shows the exterior of the front of the heart of a mammal. It has been positioned to begin a dissection. Okay, so over here, okay, now B and C is pretty tricky to find out. However, thankfully, they didn't ask you about it, because usually the heart, if it's not cut by a butcher or something, there's going to be other tubes going out of it. So I reckon that one tube is the aorta and the other tube is probably the vena cava. Okay, so the atrium is usually located in the top. Okay, we have our atriums over here. And then of course we have our ventricles over here. So A is going to be the atrium, and over here is where the ventricles meet. This is the apex of the heart, which is over here. It's usually the low bit, so it's called the apex. We have coronary blood vessels, which are located on the outside of the heart. Okay, so the apex is going to be located in E, and then an atrium is going to be A. The place where the right and left ventricles meet is going to be F. And the coronary blood vessel is going to be D. Okay, The photograph shows the inside of the left side of the heart. It was cut open. Okay, draw a line on photograph 1 to show where the cut was made. Okay, so when we had the picture of the heart on the previous slide, this was our heart. Basically, it looked like that. The cut is going to be made over here because our ventricles meet over here. So if we cut over here, it wouldn't make sense. You need to cut a little bit below it so you can see. So if you go on the previous slide, you're going to see a red box like this. This is where the cut was made. Draw a plan of the structures within the rectangle on photograph 2. Label the tendons, cords, heartstrings, and the cut surface of the muscular wall of the ventricle. Okay. So over here is the picture of the heart, and just simply draw it over here using pencil, of course. No shading. So no shading. And of course, when you're labeling a diagram, always label using a simple line. Do not put an arrow. So this is wrong. Just make a line and then write down what you want to write. Okay. The diagram shows a healthy heart and one showing a heart condition called DCM. Over here we have the healthy heart, over here we have the heart DCM. Now there are several things that you can notice. First of all, the septum over here is much thicker and healthier. The septum with the heart with DCM is very thin. Okay. Also, the heart walls over here are very thin and weak. The heart walls in the healthy heart are very thick and strong. Okay. Now they both have ventricles, they both have atriums, they both have valves. Okay. Compare and contrast the structures of these two hearts. Well, first of all, both hearts have ventricles and atriums. Now, hopefully, if you get a question like this, please do not overcomplicate it and think, oh, they're going to say that ventricles and atriums are wrong because it's too simple of an answer. No. Sometimes they will give you very simple answers. Sometimes they will give you very complicated answers or questions. But it's just most important that you do not overcomplicate, and sometimes it's better to not think that hard on the question, okay? Because sometimes I used to think extremely hard on one question when the answer was very straightforward. So now let's talk about the differences. The septum is thinner in the DCM heart, and the wall is thinner in the DCM heart. Therefore, we covered 4 points, and we gained the 4 marks. Okay. A post-mortem study was made on 68 hearts. Of these, 48 hearts with DCM and 20 were healthy hearts. Okay, so there's a comparison between the health of hearts. So we have hearts that are ill and hearts that are healthy. The diagram shows the measurements labeled 1 to 4 that were made from sections of the left ventricles of hearts with DCM and those of healthy hearts. State the independent variable in this study. So the independent variable is what you are in charge of. So the scientists, they were in charge of choosing the health of the heart or the type of the heart. So they either chose a heart with DCM or a healthy heart. Therefore, the answer is going to be the health of the hearts or the type of the heart used. Now, double I, when added together, these measurements give the full ventricle circumference. So we have the healthy heart and the heart with DCM. Okay. Measurement 1 has a huge difference of 1.93. How did I get this value? I simply minus 3.81 minus 1.88, and I did that for the whole table. Now calculate the percentage increase in the left ventricle circumference shown to be caused by DCM. Give your answer to 3 significant figures. Okay, so first of all, we're going to minus 20.36 minus 13.26, which gives us 7.10, okay? Because we're calculating the percentage increase in the left ventricle circumference, okay? And the data is given over here. This is the mean and total circumference, okay? And then we're going to do 7.10 divided by 13.28, okay? Because you want to find the increase. Okay, then we're going to times by 100 to find the percentage. Therefore, it is 53.5%, and that's one significant figure, two significant figure, three significant figures. Okay, now the final question. The mean circumference of hearts with DCM is larger than the mean circumference of healthy hearts. Okay, because remember, it's much thinner, therefore the walls are much thinner, so the heart is going to be much wider because there's no muscle holding it together. So discuss the relative effects of the changes in the four individual measurements on the overall increase in circumference. Well, first of all, the biggest change is shown in measurement 1, which is 1.93. The smallest change is in measurement 4, and 1, 2, and 3 change by about the same amounts. They change by 1.93, 1.89, and 1.90, so they're pretty similar. Okay, thank you so much for listening, and I hope that this video was helpful. Please subscribe. Thank you.